Yes,
No,
you are
you're
loud and
audible, ma. We
clear.
can hear you, ma.
Thank you very much. We are sorry we are starting 2 minutes behind schedule, but we have some housekeeping that we wanted to quickly sort out, and we have just succeeded in doing that right now. So thank you so much for being here. The first announcement I want to make is to thank you for your incredible participation last week and to say congratulations for making it here. We had quite a number of persons, but not everybody did the pre-test. Some people did the evaluation form, and it's only those who did the pre-test that have been invited to participate, to continue to participate. All the other people will come in for the second batch, and we'll let you know when the time for second batch comes. And so please, if you are here, kindly concentrate, because post-test is going to take place. And one important condition is that you're actually here. You started last week. You did not miss any of the classes. Today, we're having 2 classes this week, today and tomorrow, 8 p.m. tonight to 10 p.m. tonight, and 7 a.m. to 9 a.m. tomorrow. And you do not miss any of the classes to the end. Though that is a very important determinant of 2 things. 1, certificate of completion, so certificate of attendance. Without you completing, we will not be able to give you that, so it's important. 2, the token for data reimbursements, you will also not be able to get it if you do not complete. So therefore, it's very important for us to put in our best and then to make sure that we stay to the very end. So in a way of opening, that is it. In a way of recapping, last week we actually had a fantastic session with Dr. Ezekiah Issa, where we looked at overview and pathophysiology and clinical features of sickle cell disease. He took us through what sickle cell disease is as a genetic disease worldwide. He explained all the clinical manifestations that we could think about and gave us all the variants that we have. Then he took us through the pathophysiology of sickle cell disease, after which we explored and we dive deep into the pathophysiology to give us information about the potential factors in cell-cell interaction, where he explained the endothelium red blood cells interaction, where he gave us the gist about adherence. And then he took us to red blood cell membrane changes in phase 2, which is hemostasis. And then we went to the B part, where he explained into details coagulant protein production. There he took us through tissue platelet, tissue factors, protein C and S, platelets, and so on and so forth. And he showed us diagram that exemplify the clinical features, major among which is crisis, vaso-occlusive crisis, pain, swelling, hemolytic crisis, jaundice, sequestration, and so on and so forth. And then we went into laboratory investigation, majorly sickling test and solubility test. Then he gave us quite myriad of laboratory investigations, isoelectric focusing, hemoglobin electrophoresis, laboratory tests, HPLC treatments. And the most intriguing part of it was the question and answer time. We had plenty of time to deal with lots and lots of questions. Time will definitely not permit me to take us through some of the questions, that we had. It's awesome. And we are so delighted tonight that we have another erudite scholar, Professor Osei Akoto, who is going to be taking us through quite a number of information about what we need to know. I'm going to briefly introduce him to us. And Professor Osei Akoto is a general pediatrician. I'm going to take off my slide so that he can start sharing. I have 10 minutes. I've spent 7 minutes, remaining 3 minutes. In these 3 minutes, I will stop sharing so that he can be preparing his own slides. And I'm just going to be introducing Professor Osei Akoto. Professor Osei Akoto is a general pediatrician with interest in hematology, especially focusing on sickle cell disease. He is a professor of child health with the School of Medical Sciences, Kwame Nkrumah University of Science and Technology, Ghana, as well as a consultant pediatrician at the Komfo Anokie Teaching Hospital, Kumasi, Ghana. Professor Alex Osei Akoto is the chairman of the Technical Working Group, National Sickle Cell Disease Strategy, Ghana, and the principal investigator for the Patient-Centered Sickle Cell Disease Management in Sub-Saharan Africa, PACTS, Ghana site. Please, I'd like him to be sharing his slide while this is going on. We have 2 minutes more. I'm still on introduction of Professor Alex Osei Akoto. And I said he served as clinical coordinator of the newborn screening program for sickle cell disease, and also as the head of the pediatric sickle cell clinic and head of the pediatric hematology unit for many years. Professor Alex Osei Akoto has worked in the sickle cell clinical program in the hospital since 1993. Wow. He has contributed to the success of the newborn screening programs in the hospital and beyond. Again, he has served as a member of the Technical Advisory Committee, Newborn Screening Program for Sickle Cell Disease, Ministry of Health, Ghana, and Ghana Health Service in the early 2000s. He has led sickle cell-related research studies and co-authored many publications. He has served as the principal investigator for the Sickle Pan-African Research Consortium, SPACO 1, between 2017 and 2021. And currently, he is the principal investigator for the GBT 131, 132, and 133 clinical trials for the Ghana site. Dear participants, I am so delighted to let you know that we have a big fish in the house tonight, and we are going to drink from the fountain of knowledge from this erudite professor, Professor Alex Osei Akoto. You are welcome. Please join me in welcoming him as he takes us through sickle cell disease, basic genetics, inheritance, and risk calculation. Thank you very much, sir, and welcome. Over to you. It is 10 minutes past 8 exactly. The floor is yours. If the PowerPoint can be put on slideshow. Hello, we can hear you. If it can be put on slideshow, I can hear you. Thank you. You're welcome. If it can be on slideshow, man. Okay, fine. So you can go ahead, please.
Thank you very much, Prof. And thank you for the organizers for inviting me to speak this evening on the topic, Basic Genetics, Inheritance, and Risk Calculation in Sickle Cell Disease. As said earlier, I understand last week I was absent, but as I heard Prof. Preska say, there was a lot of enthusiasm and information shared by the first teacher in the first class. And so without wasting time, as already introduced, I'm a general pediatrician. I have no conflict of interest in this presentation. And I must say that I'm not a geneticist. And therefore, whatever we are going to discuss will be just general principles and the things that are well known to us. Our objectives will be to a quick history about sickle cell disease and its etiology. Then we'll look at some genetics concerning sickle cell disease, inheritance patterns, and then look at calculation of risks. So I'm sure from last week, we were introduced to the definition for sickle cell disease, so I do not have to belabor the point. Just to say that it is a generic term. Sickle cell disease is a generic term that refers to a condition in which an individual has two abnormal genes, hemoglobin genes, at least one of which is hemoglobin S. And the whole symptomatology and pathology relating to the disease is from what is known as the sickling phenomenon. So by way of history, I know in Africa, it is said that our people knew sickle cell for ages. And so you go through many areas in West Africa or in Africa, especially in Ghana, that I know much of. We find a lot of names, dialects that refer to sickle cell disease in different parts of our countries and our tribal areas. But historically, it is written that in 1910, Dr. Henrich in Chicago was the first person to discover, in brackets, sickle cell disease, that is sickle red cells, when he attended to a Grenadian dental student. And for the first time, this hematology doctor was seeing abnormally shaped red cells and named them sickled red cells. In 1949, Pauling and his compatriots or his lab workers were the first to describe the molecular basis for sickle cell disease. And it so happens that sickle cell disease is the first disease to be described in its molecular basis. So much is known about sickle cell disease since the 1949. And then in 1956, Ingram in his lab and colleagues in Germany discovered that the whole problem of sickle cell disease is as a result of a single point mutation for which the disease has gained its notoriety for many years. And on and on and on and on, fast track to 2008, the United Nations and WHO declared sickle cell disease as a disease of public health importance, such that countries should look at it well because it has been neglected for so long. So I'm showing this graph to show how the sickle gene has traveled around the globe, because it is known that sickle cell disease is a disease of Black people like you and I listening to me, people of African descent from the Mediterranean and Indian areas. But so the origins, 2000 BC, sickle cell was noted in West and Central African regions. But there have been migrations along the line. And when people travel, they go with their dresses, their luggages, but they also go with their genomes or their genes. So the gene has traveled around the world, and so sickle cell disease currently is known all around the world. So even in Australia, where sickle cell disease was not known, but in the last 10, 20 years, because of migration, You go to Australia and you find sickle cell clinics. This picture is showing what is known as the molecular origins of sickle cell disease, or the gene. There are 5 clearly distinct things called haplotype. There are 5 of them: Senegal, Benin, Bantu, Cameroon, and India or Saudi. So Senegal, the Bantu, Cameroon, all in Africa, and then in Indian regions. And in the context of sickle cell disease, a haplotype refers to a specific group of DNA variants or alleles, which are inherited together on a single chromosome, particularly near the beta globin gene cluster on chromosome 11, where the sickle cell occurs, mutation occurs. And the 5 haplotypes that I've mentioned, they have a significance in relation to the disease severity. So if someone has the Benin haplotype, which is in West Africa, there is a correlation between the amount of fetal hemoglobin levels in such patients. And so for those with Benin haplotype, they have moderate fetal hemoglobin, and therefore the disease appears to be moderate. For those with Bantu, that is in the Central African Republic, they have low hemoglobin F, so they tend to have severe disease. Those who have the Senegal haplotype, also from West Africa, tend to have high hemoglobin F, that is fetal hemoglobin, and therefore they tend to have milder disease. The Cameroon haplotype has an intermediate amount of hemoglobin F. And so the disease is variable. And then the Arab Indian haplotype has very high HbF, making the disease milder, all things being equal. In fact, it is known that the presence of fetal hemoglobin kind of ameliorates the disease severity, because it does so many things to the sickle cell. The red cell does not allow it to sickle, and therefore many of the complications that we see also do not occur. And I'm sure all of us have heard about the use of hydroxyurea in the management of sickle cell disease. That drug kind of increases hemoglobin F production such that the disease severity is minimized. Now, genetics is the study of genes and genetic variation and heredity, and it explores how traits are passed on from parents to offspring. And these traits usually influence an organism or a person's characteristics, including physical appearance, susceptibility to certain diseases, and even to certain aspects of our behavior. And these things we get from our parents. So that is what genetics is all about. And we all recall that the father of genetics, Gregor Mendel, studied a lot of genetics and gave us a lot of insight into how this thing comes about or play out. So what is a gene? It's a material that controls which traits are expressed in a person or an organism. And usually the genes come in pairs, and the offspring of parents usually receive one copy each from the parent. So one copy from a father, one copy from mother. And genes are made of DNA, which also help us to make proteins and other things that determine the characteristics that we inherit. The genes are usually the units of inheritance, and they code for a pair of contrasting traits, which are known as alleles. And the given set of alleles at a locus or cluster of loci on a chromosome is referred to as a haplotype, as earlier described, that we have 5 of such in relation to sickle cell around the world. The genotype of a person is a set of alleles that make up his or her genetic constitution. So we've heard of genome and all that, and we are told that there are over 25,000 to 35,000 genes in the human body. The phenotype is the observable expression of a genotype. So what we see of the person, appearance, and all that is the phenotype, whilst what is in there that is bringing out the phenotype is the genotype. Now we talk about mutation, which is a problem in the DNA sequence. DNA is structured that some amino acids, they form a chain called sequence. And sometimes something may happen to a chain or one amino acid or many of them and change the natural thing to give a problem. And that is what we are talking about as mutation, a change in the DNA sequence. It can be just one amino acid or 2 or more, or even a break of a certain amount of DNA. And it can be spontaneous or induced by external factors. And usually mutations contribute to genetic diversity and disease. And so in our talk this evening about sickle cell disease, mutation is the cause for sickle cell disease as we know it. So it may, as already said, it may occur spontaneously, or it may be induced by external factors, such as radiation, chemicals, UV lights, X-rays, and some other drugs. And there are many mutations that can occur. There are about 7 of them listed. We have what we call point mutation, which occurs in a single nucleotide. That is, a single nucleotide, amino acid may be changed. Here there are 3 types of it, subtypes. We have the silent mutation, in which there's no change in the amino acid. Then there's a missense mutation, which is changing one amino acid to another. And here I've colored this red because this is what happens in sickle cell disease. Then we have nonsense mutation. Then the second type of mutation is insertion, where one or more nucleotides or amino acids are added to the DNA sequence. Then we have deletion, which is one or more nucleotides removed from a DNA sequence. You may have a frame shift mutation, which may be an insertion or deletion that could shift the reading frame, changing every amino acid downstream. And this one usually results in a non-functional protein being produced. And we have duplication, which is a segment of DNA copied and inserted into the genome. Then we have inversion, where a section of DNA may be cut out, reversed, or reinserted. And then we can have translocation, where a piece of DNA breaks off from one chromosome to attach itself to another chromosome. Effects of mutation. The effect of mutation can happen to the protein function. And here, when mutation occurs, there can be loss of function of the protein, either by way of reduction of its activity or even elimination, complete elimination of the protein activity. There could be also gain of function, where protein may gain a new or enhanced function. And there may be dominant negative, where the mutant protein interferes with normal function. Another effect that can happen is phenotypic effects. So here, there could be beneficial effects where there could be improved fitness of the mutation occurring. And here, I'll refer you to check what is known as balanced polymorphism between sickle cell disease and malaria, where it is known that the sickle cell gene has flourished in the African subcontinent because of malaria, because it is known that the AS gene kind of, as it were, supports malaria, where people with AS usually are able to withstand malaria more than sickle cell disease or AA. And so there's a benefit of that. There may be harmful effects from mutation on the phenotype. So the molecular abnormalities in sickle cell disease. The sickle cell hemoglobin S results from a single point mutation during the synthesis of hemoglobin. And there is a substitution of one amino acid valine for glutamic acid at position 6 in the beta chain, giving rise to hemoglobin S. And then we could also have lysine replacing glutamic acid at the same position 6, resulting in hemoglobin C. So these two hemoglobins, S and C, we know them to cause sickle cell disease. And as explained earlier, the two abnormal genes that somebody would inherit from parents, if one is S and the other is S, given a double S, then the person will have sickle cell disease, SS disease. If you receive S and C from your parents, then you get hemoglobin SC, also which is disease. And this mutation occurs on chromosome 11. So this is a diagrammatic representation of the beta hemoglobin, where there are 146 amino acids. And for the normal hemoglobin at position 6, we have glutamic acid. But for the sickle hemoglobin, we have valine taking the place of glutamic acid. So these are the biochemical representation of GAC to GTG for the DNA, giving rise to hemoglobin S. So So in the same way as said earlier, if lysine replaces glutamic acid at the same position 6 on the beta globin chain, then we get hemoglobin C. So sickle cell disease, the inheritance follows what we call the simple Mendelian principle. And we'll take some time to look at this. So here I have AS, that is an individual with hemoglobin A and S getting married to another individual also with hemoglobin A and S. They could give rise to children AS. So I'll take some time to explain these ones. So we've said that the normal hemoglobin has glutamic acid at position 6. The sickle hemoglobin S has valine replacing the glutamic acid, for hemoglobin C, lysine replacing glutamic acid. And these point mutations give all the problems that are associated with sickle cell disease, the clinical problems that are associated with sickle cell disease. So the types, if somebody has hemoglobin AA, is normal, there's no disease. If somebody carries hemoglobin AS, we say that the person is a carrier or he has the trait, and usually such a person is asymptomatic. If a person has hemoglobin SS, then that person has sickle cell disease, sickle cell anemia. And here we have the full clinical sickle cell disease occurring. And also, if somebody has hemoglobin S and C, you also have hemoglobin SC disease. That is also sickle cell disease. And also, somebody with hemoglobin S and then beta thalassemia. These are variants of sickle cell disease of variable severity. The inheritance follows what is known as autosomal recessive pattern. And here, what we are saying is that both parents must pass on the hemoglobin S gene for a child to have sickle cell disease. That is to say, if somebody is going to have sickle cell disease, SS disease, then both parents, mother and father, each one of them should pass on hemoglobin S to the child. If it is only one, that is only one parent passing on only just S, the person becomes a carrier. But if both pass on S, then we have two genes, then the child will have the disease. So here we can use the Punnett square for example to show how parents can give offsprings to show whether they have normal hemoglobin, a carrier state, or a disease state. So I'll move on to so let's look at the inheritance pattern very well here with examples. Here, I have a father who is AS and a mother who is also AS. If they decide to make a baby, the mother can give her A, the father too can give his A, and the child would be AA. So here, both parents are giving of their A's to the child. So the child will be AA. In another scenario, if the mother still gave an A, and the father gave his S gene, then we have a resultant AS, so the child becomes AS, and this is a carrier state. This is not a disease state, but the child will be labeled as a carrier, so that in future he too can pass on these ones. What if this time both parents gave out the S gene? So father gave S, and mother too has given the S. Here we have a child that has both SS from both parents. So this child has sickle cell SS disease. What we must realize that we have shown 4 outcomes, but in the course that we are going through genetic counseling, you may come across a couple that will come to you and say, you said that if I'm going to give birth to 4 children, one of them could be SS. So then I should try the first one, second one, then I'll stop. Here, what we're going to say is that for each and every time that they make a baby, one of these 4 outcomes could be the result, and not necessarily, we can't pick and choose, so to speak. So each time they make a baby, if they're going to have 4, all the children could be AA, provided that both parents at each time are giving their A's. Each time the baby could be an AS, provided one of them give A, the other give S. And the other scenario is that the child can be SS each time. So here, in terms of frequency or probability, we have 1 out of 4 being AA, 2 out of the 4 being AS, and 1 out of the 4 being SS. So 25% chance that the child could be AA, another 25% that the child could be SS, then 50% chance that the child could be AS. And we are saying that each and every time they make a baby, and not necessarily that they can move from AA to AS, and before they get to SS. No. I want to draw your attention to one important fact that we have mentioned, that sickle cell disease also occurs when one parent is AC, the other parent is AS, and the one who has AC could give the C to the child, the one who is AS could give the S to the child, making SC, and SC is a disease. And we need to be careful to look at this scenario very well, because understand last week when Dr. Hezekiah gave the talk, he must have said something about the sickling test, where AC would usually come out as a negative test. Only AS or SS or SC would come as a positive test. So if you're just using that test, you could easily be taking the fact that, if you are sickling negative, then you are fine. You could go and marry any other person. So If a father is AC and mother AS, in the same Mendelian inheritance pattern, we can have a child coming out as AA, if the father this time gave a C and the mother gave an A, we can have the child coming out as AC. If father gave his C and mother gave her S, then we have a child with SC who has disease. And I'm saying that it is very important we should take note of this. There have been some bitter experiences in my practice where I've met couples who have sickle cell SC children, and it's gone very bad. The result has gone very bad because the one with AC didn't know. Recently, about 2 years ago, I met a young couple who were getting ready to marry, and the lady knew she was AS right from junior high school. Met a young man at the university. They became friends. And when she asked, the young man said, oh, she was sick, she was negative. So they became friends for a long time. Finished university, gone into employment, and then they now wanted to come together as husband and wife. And then they were asked to go and check their sickle cell status. It came out that the young man was AC and the lady was still her AS. So this is a scenario that if you don't take care, you can have on your hands. So in like manner that we discussed the ASAS, each and every time that they make a baby, we can have one of these. One AA, one being AS, one being AC, then one being SC. And the SC, we are saying that is a disease. So we should take note of that. For risk calculation of sickle cell inheritance, primarily we refer to estimating the likelihood that an individual will inherit or pass on the disease based on genetic inheritance patterns. And in sickle cell disease, we have said that it is an autosomal recessive condition, meaning that both copies of the hemoglobin beta gene must be affected for a person to have the disease. So we look at some calculations. But we can use different methods. Some of them will be beyond us here, but the one that we can easily use is the Punnett square, which is usually used for individual inheritance, the ones that we have just gone through, where an AS father meets AS mother, and then we've calculated 1 in 4 chance that the child could be AA, 50% of the chances the child could be AS, and then 25% of the chance that it could be SS. Then there's this mathematical one, which is Hardy-Weinberg, which is usually used for population level risks. We have the Bayesian risk method of calculation, empirical risk method, which is usually used for family history cases. And then these days, we are told that there are softwares that can be used for modeling and other things. So using the PANET, which is easier and for us at our level, we can use the parent genotypes to estimate the risk per child. And as we said, if one parent is AS, the other is AS, we have shown that 25% could be AA, 50% could be AS, 25% could be SS. So this is a very simple and commonly used in genetic counseling practice. Then we have the Wendenberg, which are usually used for public health planning. The mathematicians among us may be able to, as it were, do us the honors by going through the mathematics of it using this equation. In the Hardy-Weinberg equation, P squared plus Q squared plus 2PQ is equal to 1. That is, it said that for genetic mutation, most of the time, in this genetic inheritance, until something very bad happens, they are usually in equilibrium and things do not change much. So the P is for the frequency of the dominant allele and Q is the frequency of the recessive allele for a gene. And usually the P squared represents AA, 2PQ represents AS, and Q2 will represent SS. So I have an example here that I want people to go back and work it out. I know there are a lot of mathematicians among us. So as homework, we can play the game. I was very afraid of mathematics in school, so I shudder to look at it. So the maths people, please work it out. If somebody can work it out now and tell us the answer, we'll be happy. The Bayesian risk estimation combines prior probability and test results. And usually, as I said earlier, it's based on family history or career screening in the general population. And it is also used in genetic counseling and prenatal diagnostics. The empirical risk estimation is based on observed patterns in family pedigrees. But this one may vary based on penetrance or unknown modifiers. So this may not be applicable in many cases in what we are discussing, except that for recurrence risk in families with one child with sickle cell disease, we can use this to work it out. Then, as we said, these days, many of the things are becoming digital. So we can use tools like the gene counselor, PANET calculators, in order to do modeling. So to go back, we have said that AA is a normal situation, no S gene, and therefore a person is normal. It doesn't have any disease. AS is a carrier state, sickle cell trait, and it must be emphasized that in our part of the world, West Africa, the sickle cell trait is very high, quoted as between 20 to 30% of the general population. We have seen that SS is sickle cell disease. And we have also mentioned that sickle cell disease is inherited in an autosomal recessive pattern where each parent will have to offer one of the sickle genes to the child before the disease can be expressed. So some examples, we have seen this one, the inheritance risk AS, we've seen that one. But if we have an SS parent getting married to an AS parent, that is SS to a carrier, then in that case, we have a situation where 50% chance that the child could be a carrier, and then 50% that the child could be SS. So the risk will be 50% in such a situation. That is to say, if an SS parent gets married to an AS parent, then they have a 50% chance that they will give birth to a child with sickle cell disease, SS. If somebody with sickle cell SS gets married to a normal person, AA, then there's a 100% chance that all the children will be AS, that is carriers, and the risk of disease will be 0%. If we have an AS person marrying to an AA, that is the AA, a carrier, getting married to a normal person, here too we have 50% chance of AA, this normal children or normal child, 50% chance that the child will be AS, and 0% chance that a child SS could be produced. So risk is 0%. So same examples being shown here. So by way of testing whether we have understood these, AS, the risk we have mentioned at 25%, SS, AA, all the children will be, is it AA, is it SS, is it AS, or none? And we have said that it will be AS. And which combination will give no risk of SCD in this expression. So AS marrying AS, SS marrying an AS, AS marrying an AA, SS marrying an SS. And we have said that we have AS and AA getting married and not producing a child with sickle cell disease. So the same thing is expressed in this table, each parent passing on one of their two hemoglobin genes to their children, parent 1, parent 2, AA marrying AA marrying AS, AA marrying SS, and on to SS marrying SS. And if an SS marries an SS, then we know that all the children will be SS. And this, for the sake of prevention and control of sickle cell disease, we want to guard against this. So there some relevance of genetics and inheritance, as we know, with regard to sickle cell disease. We have known that sickle cell disease has helped us to understand genetics and molecular basis of diseases, because it was the first disease that gave us insight into it. And it has also helped us to know inheritance pattern, how we inherit the disease from our parents, and therefore offering us the basis for genetic counseling of individuals in order to make informed decisions and choices in life. It has also offered us insights into how best to prevent and control sickle cell disease. Because as I said, if somebody is SS going to marry another person SS, that will mean that all the children, if they get any, would be SS, and we don't want to do that. And even sickle cell disease in genetics has helped us to understand many of the things with regard to the management of the disease itself. Genetics has even given us curative alternatives to management. We know that currently methods of cure in sickle cell disease revolve around stem cell transplantation or bone marrow transplantation. And what is on the block now, gene editing. And these are all based on genetics. So genetics is very relevant in sickle cell disease. So as I mentioned, gene editing and genetics, I want to take some 2-3 minutes to look at gene editing, which is currently the boy on the block that people are looking at. Gene editing is a precise alteration of the DNA in living organisms, and in man, and especially with sickle cell disease, we are looking at using this tool to, as it were, alter the abnormal gene, that is the position, the mutation point, the position 6, where valine has taken the place of glutamic acid, to alter it so that we can change the disease into a normal one. And it includes either adding or removing or even modifying the DNA sequence. And there are some 3 tools that are being used currently: CRISPR-Cas9, Talens, zinc finger nucleases. And all these have applications in medicine, in agriculture, and then in research. And in medicine, what is driving it is research. And currently, there's a lot of research around gene editing in sickle cell disease. So we have said that sickle cell disease has just a point mutation resulting in the abnormal hemoglobin. And so gene editing targets this mutation to correct or compensate for it. And how it does it? It repairs, CRISPR-Cas9 repairs the faulty hemoglobin beta gene in stem cells. And then the edited stem cells are reinfused into the patient. So the patient's own cells are edited and then they are reinfused. So it is said that there appears not to be too much of problems with regard to graft versus host, where you use bone marrow transplantation or stem cell transplantation. And this leads to the production of normal hemoglobin A. And how it does it is that it repairs the faulty gene, and the Cas9 is said to be like a scissors that is projected and gets to the point where the problem is, and then it will kind of take it off so that the repair can be done. Then there's a second strategy where hemoglobin F, which does not allow sickling to occur and therefore protects people from severe disease in sickle cell disease. You know, in the adult, the production of fetal hemoglobin is stopped. After birth, it is stopped by age 3 to 6 months. More than 95% of fetal hemoglobin has been switched off. But in this instance, it is known that that gene that suppresses the hemoglobin F production in adults can be re-engineered or silenced, so to speak, so that production of hemoglobin F can be reboosted, and that people with sickle cell disease will produce a lot of hemoglobin F to, as it will, help with the disease management. So that's it. And the clinical significance here is that gene editing offers potential cure of the disease and not just merely the symptom control. And there are current trials ongoing, many of which are showing high success with crisis-free outcomes. It uses patients' own cells and avoids rejection. And I was happy to see one young man who has undergone gene editing and is currently disease-free. And he was at the Abuja conference 2-3 weeks ago, and he shared his experience. So gene editing provides those two powerful strategies mentioned. And people are saying it is the way to go, but there are a lot of ethical issues and also accessibility issues that need to be considered. It is said that even though people are saying that it is easy and can be cheap, but currently we are told that it is very expensive. I don't know the cost anyway. And so for us in Africa, probably it will take us many months, years to come before we are able to get there. But it is currently being done elsewhere. So this is some more information about the gene therapy thing. So how it works and the full name for CRISPR stated there. So on that note, I want to say thank you for listening to me. That is my last slide. And I would hand over to Professor Priska and her team at the Anchor site to carry on with the rest of the session. Thank you very much.
Thank you so much. Fantastic. We're so grateful. Done incredibly well. There are so many questions. And I'm going to turn it over to Dr. Oriaku to moderate the Q&A session. And we just encourage you to take a glass of water and relax back a bit. Thank you so much. You've been speaking for quite a long time so that we can take questions and comments from the chat. Over to you, Dr. Oriaku.
Thank you, Prof. Thank you, Professor Alex-Okuti, for the interesting lecture. It was quite educative, insightful. Thank you for making it interesting too. I remember back then in medical school, genetics are not that wonderfully presented. Sometimes it looks abstract, but you just split it down to our level, making it look as if it's just reciting ABC and numbers 1 to 10. Thank you so much for the lecture. Our first question, sir. Somebody wants to know, where does HB, SS, SC, and any other form of sickle cell disease fall, whether it's genotype or phenotype?
SS, SC.
And any other form of sickle cell disease, where do they fall? Is it a genotype or a phenotype?
That is a genotype. The
Right,
phenotype is usually the outward appearance of the trait. So I think I explained it earlier that yes, when you do your labs and it shows that you are SS, that is your genotype. But the phenotype is usually referring to the outward appearance characteristics, the severity of the disease, and all that go with it. So that's the simple answer I can give.
Okay. We have more. Someone wants to know whether, well, which you already know, whether parents with AA and AS can give birth to a child with a HBSS. That's our second question.
Yeah, that's interesting. I think that, yes, I explained that so many times that when an AA gets married to an AS, you will not get SS. But you see, interestingly, there's a condition where some people have a thalassemic trait and they are labeled as AA, right?
Yes.
So if such a person ordinarily with HB electrophoresis would be labeled as an AA, if he gets married to an AS person and transmits the thalassemic trait to the child, and the other person, that is the other parent, has transmitted the S, then you have sickle cell disease, that is sickle cell thalassemia. And most of them would appear clinically as SS. Many years ago, I had an experience of a family. The woman was AA, the man AS. The man, obviously, we knew he came from a family that has sickle cell disease. And they had two boys. And the boys appeared as SS. They had multiple transfusions. And along the line, the man unfortunately decided to ask for divorce from the woman. And his reason was that the woman had gone to another person. That is why she had given birth to these children. It was a difficult question for us because at that time we didn't have enough facilities to do what. And we realized that from the history, everything pointed that, oh, this is the woman with AA, the man has AS, the children had phases of sickle cell disease a bit, but it was a dilemma. Fortunately, I spoke to Professor Imelda Bates. I'm sure many of us on the call may know her. She's in Liverpool School of Tropical Medicine, a hematologist. And she graciously asked us to send samples to Liverpool. In fact, I've forgotten which year was it. That was many years ago anyway. And the results came back, and that showed us that, yes, the woman had thalassemic trait, and the man obviously had his own AS. So the man had given the S, the woman had given the thalassemic trait to the two boys. And they were showing these signs of sickle cell disease. So yeah, in all fairness, a true AA marrying a true AS will not give rise to SS genotype, as we have shown many times during the talk. Thank you.
Thank you, sir. This takes us to our next question. Somebody wants to know how to detect HbAc when it shows negative result during genotype screening.
Please come again.
Somebody
Hello?
once asked how to detect hemoglobin AC,
Okay.
not AS, AC,
AC.
when
Okay.
it shows negative results during genotype screening.
I don't know what a person means, but my thinking is that he wants to find out how do you demonstrate that somebody has AC. It is usually the normal HB electrophoresis or the use of HPLC or isoelectric focusing, any of the hemoglobin electrophoretic tests that will show the C. With regard to solubility test or sickling test, those ones, they become only positive when S is present. So with just the AC, it will be negative. So you are unlikely to know that that person is AC. So the best thing to do is to do hemoglobin electrophoresis to give you the actual genes, that is the genotype that a person has, either A and C or A and what. So that is what I would say.
All right, Prof. This takes us to the next one. The difference between sickle cell disease and sickle cell thalassemia.
The difference between
sickle cell disease and sickle cell thalassemia. Somebody wants to know.
All right. So sickle cell disease is, I said that when you say sickle cell disease, usually you are using an umbrella term to refer to any of the diseases, that is SS, SC, or S beta thal, in which the person, that is the patient, so to speak, has hemoglobin S and one other abnormal gene. So it could be S, so SS, or SC, or S beta thal. In sickle cell SS and SC, we are saying that it results from the mutation where glutamic acid on the beta globin chain, the 146 of them, the number 6 position instead of glutamic acid has been replaced by valine to give S or by lysine to give the C. That gives us the S and the C. But in terms of thalassemia, it usually refers to a situation where the problem is to do with the amount of the globin chain that is produced. Some are not produced, so that it ends up being an abnormal protein, and therefore when it combines with S, then you get the S beta thalassemia, and there are different versions of it. S beta thal, major and minor. Thank you.
Thank you, Prof. What is the best time for gene editing, and what is the success rate, chances rate for the procedure?
that one, I will say that my knowledge and skill does not reach there. So I'll not be able to say that.
Okay. Thank you, Prof. Another one goes, does gene editing affect the germ cell line or somatic cell line?
Yes, it is said that when the procedure is done, the cells become normal, so to speak, in terms of the person begins to produce normal cells. So he may become AA or something, because you take out the gene that is, as it were, making the person produce the S gene and replace it with a normal one. So that is the explanation I got from the reading.
All right, sir. Then somebody wants to know the importance of genetic counseling.
Genetic counseling.
Yeah.
I'm sure the person is here being trained
Because
as
of
a
that.
genetic counselor.
Yes, sir.
So
At the end of
I was thinking that probably you would have read a lot about it, but there's a lot of importance to it. And you know,
yes, sir.
we have, as I said, I made mention of the fact that in our countries in West Africa, we have a career rate of between 20, the least career rate will be like 15, but between 20 and 30. In Ghana, it's about 20, 25. In Nigeria, it's about 30. So that is of the whole population, 30% of us, so to speak. So if we do not have people to be able to counsel our young and upcoming ones, and also if people don't know their status, they will go marry anybody, and before you are aware, they would have produced sickle cell patients. And in fact, genetic counseling is not even only for sickle cell disease. It could be for all other conditions that are related to genetic issues. So it is very important for us to have people well trained, so to speak, in genetic counseling, so that they'll be able to share information, give counseling and advice to people to make informed decisions about how to, that is for even for those who have the disease, how to, as it were, run their lives, and those who are going into potential marriages to be able to, as it were, have knowledge about what they are going in for, so that they don't, as it were, have problems. I can share an experience with you. Many years ago, I had a mother with two sickle cell SS disease children, very severe ones. This one will be in hospital this week, the next week, the other one will come. So this mother was, very stressed. Unfortunately, he said the husband was not supportive, so she left the marriage. Some 5-6 years down the line, that woman comes back to clinic with a new child with sickle cell disease. I asked, with all the information you had, said, Doctor, I did not let the man go and check. You see, so such are people who, if they have real knowledge and information and have the needed information, and they understand and can make decisions about themselves and their lives in respect to the children they are going to give birth to, I think that we may not have big problems as we have on our hands with many of our people and families, so to speak, having sickle cell disease warriors and with problems. And many of our resources are unable to, as it were, help us manage them the way we want. Thank you.
Thank you, Prof. There are so many questions on if gene editing leads to cure. Can somebody that was previously with hemoglobin SS after gene editing, HbAA, go ahead to marry somebody
Yes.
with HbSS after gene editing?
I want to believe yes, that yes. If he is cured and has AA and is always going to give an A, then he or she could, I'm using could advisedly. Well, I don't have all the information yet. So, and you know, it's a new ground. So not all is known yet. But I believe that with the current knowledge of an AA person who could get married to an AS or even SS and producing AS, or that is to say a zero sickle cell SS patient, yes, then that should hold.
Thank you, sir. What's the difference in the severity of SS and SC crisis?
And the difference between
SS and SC.
and SC.
Yes, in the
Okay.
severity.
Okay. It is known that SS, all things being equal, SS is referred to as the severer of the forms of the disease. SC can be moderate or mild. That is how it is referred to. But we have instances where some factors come into play that we have some patients who are known SC, but they tend to have very bad events. But generally, it is known that SS is severer than SC. Is that okay?
Yes, the difference between
Okay.
the severity of their crisis, the SS and the SC, sir.
So I'm saying that the SS usually tend to give severer problems
All right,
than
sir.
the SC patients. You know, in my practice, I've come to see about 3, 4, 5, 6 patients who were diagnosed of SC disease when they were about 46. In fact, somebody was 49 years before a diagnosis of sickle cell was made in the person. And he said all along his life, he had not had any problems until one day he went to work and had to pick up something, had a waste something. And so the problems, he came to the hospital, somebody said, okay, let's check your blood and see, and then it was picked up, not necessarily that person thinking that he has sickle cell, but it was picked that his sickling was positive, and therefore the electrophoresis was run, and it came out as SC. So yes, some SC people tend not to have problems, but most SS people will have problems along the line, along the continuum of life. Yes.
Thank you, sir. Is there any contradiction for
Doctor,
Okay. All right,
could you
sir.
please,
All right,
thank you. Sorry to interrupt you. Could you
Okay.
please allow Professor Nodu to contribute to the question on gene editing?
All right, ma'am.
Thank you. Over to you.
Yeah, okay. Thank you very much. Good evening, everyone. I want to thank my buddy, Professor Alex Akoto.
Hey, Obi, body. Obi, go ahead, please.
I hope we
Okay.
didn't look up.
Yes. Thank you very much. Good evening, everyone. I want to acknowledge the wealth of experience that Professor Alex has expressed, and the way he's answering the question. And I want to tell you people that we're going to put out more information about these genetic counseling and some of the other questions that are being asked, so that people can go and to that resource and read more, because we can see that that cannot be attended to in a short webinar like this. But to the question on gene editing, you will all agree that this is a new area for all of us, and we are still learning about them. Somatic gene editing is looking at differentiated cells, right? Nobody is doing germline gene editing. It's not allowed in medicine yet, where you actually affect the ability of that gene edited person to transmit the new genes to the progeny. It's not allowed. So what is being done in gene editing are the stem cells, right? Not the germline, not the ones that carry, the genetic information to the progeny. So if you have gene editing, you still have your germline genes are not touched, and they can still pass the sickle cell genes, to the children. You understand? It just makes that person okay, but that person can transmit the sickle cell gene to the progeny. You understand? So I just want to make that a clarification, because this is a new area and we're still all learning. So they are working on the reversing the switch for the fetal hemoglobin, and then some are taking out the abnormal hemoglobin and putting a new hemoglobin A. You understand? So it's a very, very new area, but the germline is not touched. So that person can enjoy a good life and still pass the sickle cell gene to the progeny. Thank you. Over.
Thank you very much, Obi, for saving me. As I said, I didn't have enough knowledge in that field.
We
Because
are all learning. We are all learning. It's a new area. And please, no questions on gene editing. This is genetic counseling. Thank you.
Thank you, Professor Nodu. I appreciate, ma'am. You're welcome, ma'am.
Thank you.
So we all need to go back to the resources that will be provided and learn more, like Prof have already said. So for now, all the questions on gene editing will go back and
Please, you can ask the remaining questions that are not in gene editing.
that's what I'm trying to filter them, ma'am.
Okay, thank you.
Yes, Prof, sir. Please, when can HB electrophoresis or sickling test be done for an infant if a newborn screening test for sickle cell is not available?
Excuse me. Newborn screening for sickle cell disease.
Let me take the question again.
Okay, take
Please,
it again.
Please, when can HB electrophoresis or sickling test be done for an infant if a newborn screening test for sickle cell is not available?
Okay. There are two things muddled up in this question. HB electrophoresis and then sickling test. There are some electrophoretic tests, or what we call HB separation tests, that can be done at the newborn period. And that is what we have as newborn screening for sickle cell disease. There's HPLC, there's isoelectric focusing, and there are point of care tests that also use other methods that can be used for HBE separation during the newborn period. But what is not done is that during the newborn period, because there's a lot of fetal hemoglobin, about 90% fetal hemoglobin in the newborn, the adult hemoglobin type that the child has, let's say the S, if the person is going to have S, is not allowed by the F to be separated by the sickling, the usual sickling test. So if you do sickling test in the newborn, it will be negative because the presence of fetal hemoglobin does not allow S to sickle. So you will find the test being negative. So if you want to do sickling tests in the newborn, no, you have to wait until about 4-6 months when most of the hemoglobin F has exited from the blood of the child. Some other electrophoretic tests are also not very good in the newborn period, like the normal citrate agar gel electrophoresis that we do, many of our facilities do. They are also not good in the newborn period because in the same way, the presence of the hemoglobin F does not allow the F to be separated. So you can't use that one. Rather, some other tests are available that can be used. As I said, you can use HPLC, high liquid chromatography, powered liquid chromatography. You can use isoelectric focusing. And as I said, currently there are some point of care tests, the Gazelle and others that are readily available on the market that can be used during the newborn period. I know in Nigeria and in Ghana, some of them are available in some facilities that are being used. Probably Dr. Professor Nodu can, as it were, tell us more about what is being done in her area. Thank you.
Thank you, Prof. Can a pregnant woman detect?
Sorry, Oriako, can I say something? Let me just add a little bit, to that, because this is the problem that we are having when we do offer newborn screening using isoelectric focusing or any other, especially isoelectric focusing, and people are not used to newborn screening yet. So immediately they go and do hemoglobin electrophoresis, and they cannot get a good result. When you test a 6-week-old baby using a hemoglobin electrophoresis. So you just to understand that newborn screening is different, and before the age of 6 months, you cannot use hemoglobin electrophoresis to test if a child has a sickle cell disease. It will not give you a good response, for the reasons that Professor Alex has said. So it's important that we inform people, because once they have that newborn screening test, they start going around general hospitals, private laboratories, and they give them a different result. You understand? So we need to be able to tell people that no, before 6 months, you need specialized equipment to be able to get a real result. But now, just like Professor Alex said, there are point of care tests which don't, you can get accurate results in newborns, and the parents can even also just test themselves and see. Thank you. Over.
Thank you, Professor Nodu. We appreciate that explanation, ma'am. Thank you. Professor Alex. Professor Alex.
Yes.
Can a Can a pregnant woman dictate her child's gene before birth? If yes, can it be corrected at that stage if detected to be SC or SS?
Please come again. I didn't hear the first part.
Can
Well,
a pregnant
woman?
dictate the
Mhm.
genotype of the child if
Okay.
found to be SS or SC? Can it be changed?
Yes, a pregnant. There's what is called prenatal diagnosis for sickle cell disease, where it can be checked whether the fetus has sickle cell disease or not. And usually it is done in the first trimester of pregnancy. You can take chorionic villi or amniotic fluid and test for it. In fact, not many centers do it anyway. But when it is done, changing the did you say changing the SS or what, the genotype?
If the genotype is
Can be
detected
changed.
to be SS or
That
SC,
can it be
can it
changed?
be corrected?
Corrected.
Yeah,
For the baby, I would say that one I don't have much, but I don't think so. If the baby is already formed with SS, it cannot be changed. But as we're discussing about these new areas, I know that there are some, I don't know whether they are licensed here. There are some technologies available that are able to, as it were, sample. But even that one is done very early, quite early before implantation, where the gametes are sampled and then tested, and depending on what the decisions of the family is, they could either go for abortion or whatever. But I understand that some also will sample it and then pick up the normal gene ones. That is, if during the testing, if they are normal ones, they pick it up and then they can reimplant. It's a difficult area anyway. It's a difficult area. See, but I don't have much of that information, but I know that there's some talk about trying to, as it were, when you do prenatal diagnosis, the main thing is to find out what your child has. And if it's SS, as we say, we know that some people have gone to abort if they didn't want it, but that is in genetic counseling, you won't preach that anyway. It is the decision of the parents to do. If they keep it, fine. But changing the genotype is a difficult area. And as I was saying, in this era of genetic changes and all that, I know some people were trying to do some things like that. But as Obi said, those things, because they are new areas, regulatory authorities are not in favor of those things because people can use it for so many other things. Thank you.
Thank you, Prof. The last question. As a genetic counselor, what advice is one expected to give a couple whose genotype are both AS and that are about to marry?
Please, we
Thank you,
have
sir.
4 minutes more. We have 4 minutes more. So after this, 4 minutes.
That's the last question. That's the last question, ma'am.
Thank you. Well
that's
done.
The last question.
Okay.
Thank you, ma'am.
So for a couple who are both AS going into marriage, I think in one of my slides, I showed that genetic counseling is very, very important in these discussions we're having, because we want to, as it were, prevent and also control sickle cell disease, because we know that there's a lot of suffering and all the related problems to do with it. And so if, as a genetic counselor, what you need to do is not, that is the teaching, is not to decide for the couple, but you are to give them all the relevant information to them. And then you ask them to make their own informed choice or choices. I have had several episodes of would-be couples being referred to me because they are both AS and AS. And usually what I do is to, as it were, take them through, like we have done. AS, these are the sort of outcomes you are likely to find. And then you add about because from my practice, I know what goes on, and I'll add those things. And usually a one-time encounter is not good enough. So you give them all the information and you ask them to go back and think about it and then come back. And when sometimes they've gone and done their own research, gone to ask and all blah, 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 and they come back and they kind of come to a conclusion that no, they don't want to go on. Others, unfortunately, would say God gives, God takes, especially for our people of faith. They may go on. Unfortunately, I have two very bad cases where people of faith said they will go, but first pregnancy, SS, second pregnancy, SS, and the problem started, and then within months after the second birth, the marriage collapsed. We don't want such things, and so we want to give the couple all the information that is available to let them appreciate what is in store in future. The love only, being in love only is not what should make somebody decide to go in for marriage. And so there are a lot of things that will come into play anyway. So as genetic counselor, that is why we need this training. But as I said, you as a counselor does not have to, as it were, push the people not to marry or to marry, but you give them all the information available to you, let them make their choice. Thank you.
Thank you very much, Professor Alex. We appreciated the time, the answers you have given. It was quite interesting. Thank you. Professor Priska. Professor Priska.
Fantastic.
Thank you, ma'am.
Job. Thank you so much, Dr. Oriako, for excellent moderation, and to Professor Alex Osei Akoto. I love that name, sounding like a Yoruba name to me. And I really
That's
do.
interesting.
Very interesting.
My Yoruba friends have not told me that before.
Yes, Akoto. It sounded to me with another thing. But thank you so much, especially for the rich and robust discussion on the question and answer session. We really appreciate you. Thank you so much. We are grateful. So we're going to spend the next 14 minutes for 4 minutes for housekeeping, and we will be glad to have the participants go and rest and the housekeeping information. But before then, maybe I should say a very big thank you to Professor Obi Anadu for finding time to join. I expect her to be resting and sleeping, but somehow she has decided to join us as well. So we appreciate your distinguished presence. I want to thank all the LOC, the MPIs, the co-Is, the executive committee members who are present tonight. Thank you so, so much. We appreciate you. And I hope the resource person for tomorrow, well, she's here so she can hear us. I don't want anybody to miss out tomorrow. The time is 7 a.m. to 9 a.m. like we had last week. And we're going to have a very distinguished personality who I'm not going to release the details tonight, speak with us tomorrow. And it's going to be, exciting as well and educative. So please do join us on time. Don't sleep off. Go to bed early, set your alarms, and make sure you're in. Now, for housekeeping, I know there's been a lot of communication going on the platforms, and I want to give you the good news that we are working on how to have the second batch of this training. So for those, the half part that we have cut off is because of the reasons that we have been explaining on the WhatsApp platform. But as soon as we end with this one, and if possible before the end, but I want to suggest at the end of this, we go back to table and then put together the plan for the second batch for those who are not here. So please do not send the link that you have to anyone. It's very important that we keep this number. We don't want to go beyond the number so that it can be manageable, so that we can have time to attend to all the questions that you have. We don't want you to have questions not attended to, and so we don't want you to please to just go ahead and be sharing this link that you have tonight. Please do not share the link with anyone. If anyone has any explanation, information, let the person reach out to us and we will be available to respond to them as much as we can. But be rest assured that we're going to take care of those people who, for one reason or the other, could not join this. I know 900 and something of us registered, but please just to understand that we are going to take care of that group. And so don't bother to send link to anyone. And tomorrow morning, the time remains 7 a.m. in Nigeria and 6 a.m. in Ghana. So to our Ghana scholars, thank you so much for your incredible presence. You surprised everyone last week, and I know tomorrow is not going to be a difference. So let us try as much as possible to come in early. Our speaker, I'm sure, will be here. And I want to thank everyone. We have just 1 more minute so that we can all go and sleep and take a rest. Please go over the slides. I will send it to us tonight on the WhatsApp group, the new WhatsApp group, where all of us on this call tonight are. And then we can go through it. Thank you very much. I want to say good night, a happy night rest. And so you can leave while the LOC quickly have the remaining 10 minutes for debriefing. And then everybody can go to rest tonight. Once again, Professor Alex Osei Akoto, I want to say a very big thank you to you. We want to appreciate your time, your presence, and your very rich profile that we read. We are so grateful that you created the time, not just to prepare the slide, but to be here to make the presentation and to engage with us, the attendees, during the fantastic question and answer session. Thank you very much, and God bless. Good night, everyone.
Good night, everyone.
Good night. Thank you.
Good night.
Good night. Thank you
Good
so much.
night.
I'm expecting your presentation so I can send it to the participants. Thank you.
Okay. Obi, good night.
Good night, my dear.
Right.
Well done.
Thank you.
Mhm.
Okay, Mr. Shola, so where are you taking us to? Are we
Yeah,
migrating?
we'll meet.
Go ahead.
We'll meet on. We should go to our LOC page. The link is there. I will hand this meeting.
Yes.
Okay, wait. I want to say something.
Yeah, I'm just thinking that people should, when you're giving the housekeeping, people should restrict the questions to the topic.
And let's go to the housekeeping so that we can go
Yes.
and you can close this.
They're just asking anything that comes into their mind.
But you know that, okay, let's go to the housekeeping so that we can quickly spend the 10 minutes to talk and then we can leave. Thank you. Okay, go to LOC.